Hey guys, I'm Savitha.
I'm Grace.
I'm Akhilesh.
And today we're going to be talking about the design of novel benzimidazoles as KRAS inhibitors for cancer chemotherapy. So the cancer gene was first found in the Kirsten RAt Sarcoma virus, and it relays growth signals from outside the cell into the cell's nucleus. And thus, when it's mutated, KRAS becomes stuck while activated, so the uncontrolled cell growth and signaling causes cancerous growths. Although KRAS is minimally studied, KRAS mutations occur in approximately 25% of all human cancers. And further, this mutation occurs in more than 90% of pancreatic cancers and about 25% of lung cancers. However, KRAS is difficult to combat due to its lack of clear binding sites, making KRAS one of the most elusive targets of cancer research. To combat oncogenic KRAS, benzimidazoles have been showing promise by inhibiting the KRAS protein. Many of its derivatives also have cytotoxic effects on mutant lung cancers. The benzimidazole derivative we focused on, Nocodazole, has been known to prevent cell replication in the G2 and M phases, as well as promotes apoptosis in cancer cells, thus making it the starting point of our research. To attain the KRAS protein and the benzimidazole derivatives, the Protein Data Bank and PubChem were used. For our methods, we created potential scaffolds with the base Nocodazole on Avogadro. After making these structures, the molecules were optimized and then uploaded to Chimera for molecular docking. To set up the protein, which was the crystal structure of the GDP-bound human KRAS, we removed extraneous chains and non-standard residues. After preparing both molecules, we used the Chimera extension AutoDock Vina to dock these molecules. To receive information like binding affinity, intra and intermolecular hydrogen bonds, and Columbus surface coloring. As we continue docking, we continue to adapt to the shapes and atoms of the molecules and the binding sites as needed by adding aromatic rings, nitrogen, carbon, as well as changing the bonding orders as well.
So in our results section, on the first row is the original Nocodazole molecule. It's with a chemical formula of C14H11 and 3O3S, and had a binding affinity of -7.1. And in the docking result, you can see how it looks binded onto our 4OVE human KRAS. And then in the second row is our A1 Nocodazole derivative that we made with a chemical formula of C14H16 and 7O6S4 with a lower binding affinity of -9.8. And then you can see how our Nocodazole derivative looks docked onto the GDP-bound human KRAS. And so in conclusion, Nocodazole, a derivative of benzimidazole, is used as a primary structure in creating novel benzimidazoles to inhibit mutant KRAS for cancer chemotherapy. So from docking using AutoDock Vina, the addition of sulfur atoms to the Nocodazole has consistently created Nocodazole derivatives with a lower binding affinity, indicating a higher chance of success to target the KRAS, which is better than the original Nocodazole molecule, which had a higher binding affinity. And so our modified Nocodazole A1 sports an increased number of sulfur atoms with 2 bonded together and 4 sulfur to oxygen interactions, in addition to an increased amount of oxygen and nitrogen interactions. And there was an overall inverse correlation between the number of hydrogen bonds and binding affinity, so that when scientists in the future continue to make more Nocodazole derivatives, it would be better to create a molecule with less hydrogen bonds.
In terms of the data sets that we derived, we compared both binding affinity and hydrogen bonds, as well as binding affinity versus the number of hydroxyl, sulfhydryl, and amine groups. In terms of the binding affinity scale that we wanted to achieve, we were looking at around a -8 to a -10 scale, because as the binding affinity goes lower, the higher the biomolecular interactions. But as shown here, there are certain data points that have hit that -8 to -10 scale, but more of the data points were concentrated between the -6 and -8 scale. In terms of things we can do in the future to help continue this project, one main thing we can do is, first of all, develop these molecules in a lab and do many preclinical tests which would more accurately portray the drug's efficacy. If you guys want to look at more supplementals, you guys can scan the QR code here under references. And to finish off, we would just like to say thank you to Mr. Bra and ASDRP for giving us this wonderful opportunity, to do certain projects like this, for example. And if you guys have any other questions, please feel free to ask. Thank you.